Swayam Prabha. Digital India. Educated India.
So, welcome back to the third lecture of the 9th week. So, we have finished about cornea and about lens. So, today we are starting the retina. So, retina is one of the most well organized structure of the nervous system. Week 9, lecture 3. W9L3, it is about retina. It is also called the neural tunic. Tunic means the girls, they put a tunic. It's something like a it's a small piece of cloth. It's kind of a, it's an attire for women. You just check it out, okay? So, it's a neural structure, and I have already kind of given you the blueprint where it is lying. This is your lens, and this is your cornea and ciliary muscles out here, controlling the exposure, and here is your retina. Now, this layer, so here is the lens, here is the cornea, here is the retina. So, this layer has photoreceptor or light sensitive cells, which are called the photoreceptor cells. And the two kind of photoreceptor cells which are involved in coding for black and white images and color images are called rods and cones respectively. So, the rods are the ones which are involved in coding the intensity in a way. They are not really specific to any wavelength, whereas cones are specific to wavelengths. So, there are RGBs, there could be red cones, there could be green cones, there could be blue cones, and in lower animals like fishes and everything, they have a fourth type of cone. So, in other words, these rods and cones have molecules which are actually proteins which are sensitive to light. In other words, when the light falls on them, they respond in a very unique way. Just the way we talked about mechanosensation, you remember? The same way these cells respond to light. We talked about olfaction, where smell molecules go and bind, and it opens up cation channels. Similarly, here also when the light falls, so whatever we have done, so we talk about mechanosensitive ion channels, olfactory molecule sensitive ion channels, and now we are talking about light sensitive ion channels. So, in other words, there are proteins which could sense to different physical stimulus and code that information. So, a day may come when we may have a protein camera, because if protein can sense this, then they could be used to do anything, but in a distant future. So, for today, let us try to understand what retina is doing. So, I have talked to you about the rods and cones. Okay. Now, let me kind of put it in the perspective. If this is the part of the retina and the light is coming from this direction, so the way I drew the structure is something like this, right. Now, on the back of these cells, I told you that it is dark. So, this layer on the back of the eye is called, as I mentioned, retinal pigment epithelial layer. In short, in several textbooks, they will call it RPE layer, RPE layer. Okay. This RPE layer is something like, it is a very interesting kind of layer. This is something like this. Lot of pigmentation in these. They are kind of dark and very heavily pigmented layer which are sitting there, and it is a very tightly packed. They are very, very tightly packed. This is not, you will not see a lot of gaps out here. This layer is something like this, and very, very dark. When you, those who have dissected eyes from experimental animal, they know that this is one layer which is kind of something like this. So, this is part of that layer. These retinal pigment epithelial layer does two things. First, it provides nutrients to the rod and cone cells. Second, it prevents or supports, prevents photo damage of the rods and cone cells. So, the rods and cone cells suffers from lot of photic damage. Photic damage means the damage which caused due to light, because whenever there is an incoming radiation, it invariably, because of the intensity of the energy, could break molecules and could generate a lot of free radicals. Those free radicals may lead to oxidative stress. So, most of these rods and cones suffers from two inherent problem, rods and cones, which together is called photoreceptor cells, photoreceptor neuron. Okay. Cells, they are, since they are very specialized neurons, I am calling them photoreceptor cells / neurons. They suffers from lot of photic damage or a damage because of light, one. Second, oxidative stress. O dot, just to tell you, O2 dot, just to tell you that this is an oxidative stress. So, now these oxidative stress and all these things are partly being handled by this layer, a retinal pigment epithelial layer. This is the one which helps these cells, and third thing it does, it provides a cushioning layer for rods and cones. Okay. So, now from the back, so always remember rods and cones are sitting on the back of the eyes. Okay. Out here, now I am putting another next color which is in yellow, just to get the contrast, is your rods and cones layer. This yellow layer, what you see. This is the layer which is okay. So, now we will discuss about the second layer, which is the rods and cones. Now, the rods and cone cells are sitting on this layer in a very unique way. So, let us see the geometry of the rods and cones. The rods are like this. Okay. Why these are specialized neurons, I will come to that now. Once I draw the structure, so you have a nucleus here, you have the mitochondria is all over the place, a lot of mitochondria, and here it's a very specialized structure out here. This is a double layered structure sitting out here like this. And they really look very nice in a cell culture dish when you look at them. Okay. Now, these cells, there is something, this part what you see is called outer segment. This part what you see is called inner segment, and this is the part which is called synaptic terminal. Now, these cells, if you tilt it in the context of the retina, now this is how we are drawing the retinal structure, right? And this is the layer we are talking about. The thin yellow line what I am drawing is your retinal layer. Of course, I haven't done a mistake by not. Okay, once again. Okay, get it right. Okay. Here you have the retinal pigment epithelial layer. Right. So, okay. So now here, the way, so I have shown you this layer. Okay, they are sitting like this. So now on top of this, these cells are sitting. So, you have to kind of rotate it like this, rotate it in this direction. So, it will be something like this. When I rotate it, you have the synaptic terminals pointing out, all right. So, I have just rotated it. If I consider this as a 90-degree angle and this is 90-degree, right. So, I rotate it horizontally parallel to the plane. So, make it a 0-degree angle. So, now from 90, so initially it was like this, okay, and now I rotate it. Once I rotate it, it becomes horizontal to the plane. Okay. So, this is how it becomes after the rotation. So, these cells are sitting out here like this. They are sitting out like this. Okay. As if this part, it's extremely essential to understand, this part is as if getting cushioned by the retinal pigment epithelial cells, cell layer, because mind it, this is the part which is, I will come to the detail of this part, this is the part which is getting the maximum photic damage in this process. Okay. So, this part is the one which is needed to be exposed to the maximum care. So, okay. So, cushioned by the retinal pigment epithelial cell layer, which is, so before I proceed further about the structure of the retina, let me introduce the next cell, which is the cone cells. So, the cones on the contrary, they actually look very similar to the cones. Now, if you look at the cone cells geometry, they are more like this. Something like this, something like this. Okay. And exactly the membranous structure, what I have drawn in rod, it is just the shape is different. Okay. Out here you have the nucleus and all other cell organelles which are present there. This is the synaptic terminal. Again, you have to rotate it at an angle of from 90-degree to 0-degree. That way you can really place them. So, they are sitting like this, synaptic terminal like this. Like this. Okay. So, now coming back to the structure, synaptic terminal, this is the outer membrane, this is your inner membrane. Pretty much the same terminology happens, it's just the structure is different. Okay. So, now in the light of this, so we have talked about the anatomy of the rods and the cone cells and their position on the retina. Followed by that, there are 4 other cell layers which are to follow. So, out here, if this whole structure, so this is the synaptic zone. This is where it will contact with the next layers of cells. So, we are now the first layer, followed by these synapses on something called horizontal layers like this. Then in between there are cells which are called amacrine cells. These does not have axon. These are neurons, though they do not have axons. These are called, this is another cell layer, amacrine cells. Horizontal cells. Then there are certain neurons which is forming another layer which is called the bipolar cells. And then finally, there is a layer, a major layer of neurons, which is the final layer, and I kindly recommend you please go through any textbook or anything about the whole detailed structure, but in the light of this, which is called retinal ganglion cell layer, which is last layer. So, this layer, all the axons kind of accumulated in one place like this, and all these axons of the final layer arranged like this. So, once they arrange like this, so there is always a spot in the eye from where this all these wires are passing, okay, and that particular spot is called the blind spot of the retina or blind spot of the eye. That is one spot where, so this is that blind spot, this is the blind spot. At the blind spot, no image is being processed. Since no image is being processed there, that is one zone where it is completely devoid of any other, you really cannot see any of the photoreceptors or anything. Okay. With this background, I will close in the class. This is our third class, and the next 2 classes will be dealing with the electrical processing of the retina. Okay. Thanks a lot.